I and a research associate professor at GBHI Trinity College of Dublin. And with that, I'll turn it back to you, Augustine, and take it away. Thanks for joining.
Thank you. Thank you so much, Fadi, for your invitation. It's a great pleasure. I have great friends and colleagues in Texas, so looking forward to explore more collaborations with you. I will share the slides. Let me see.
So the share is next to the leave button. If you right corner to the upper right corner, it's share.
So I see, but I don't see how to put the. Let me, because I only can see my calendar, or there is no option to share my. Maybe here. Just a second. Sorry. Let me see. Maybe Windows. So it's asking for open Chrome. I don't know why. Oh, I need to configure. Sorry, it says that I need to configure my. Oh my goodness! Give me a second. It's
okay. Take your time.
System. So I need to put something in the system settings, but something is not working. It's like don't in a buckle. Sorry, I don't know what to do. Let me check. If I can go out. No. That's strange.
So sometimes it happens before. If you can log off, log back in, probably.
Okay.
Sometimes.
Okay. Sorry for that. I will do that.
Oh, no problem.
Can you see my screen now?
Yes.
Oh, fantastic. Sorry. It was something in the system settings that I need to change. I cannot. I hope that you can see it now.
Yeah, thank you.
Thank you. Wonderful. So thank you so much for your time. I will present some ideas about how to link resilience in brain health settings, especially considering the role of, interactions between the exposome and the whole body health. That I think that are important to understand resilience in larger settings. Those are my disclosures. And so, as I mentioned, the overall idea is why resilience is important in global settings. Why it's important also to develop the, or very long expected precision brain health, and how neurosystemic frameworks involving whole body and exposome are important to understand that. So first, I will start with this very poor definition of resilience, as a very general terms, dynamic ability to adapt and thrive in adversity that encompass biological, psychological, and social dimensions. Later, probably you will get what I am trying to bring this particular definition on that. But first of all, why we need to understand that in precision and mental brain health. So first of all, when we try to understand resilience, we can think like in genetic resilience, in immune system resilience, we can think in adaptations, self-regulations to handle stress. You can think in community resilience, you can think in social resilience. So the tricky thing is that for developing any precision metrics that should be related to resilience, to brain health, or any other domain, we really need to bring data diversity, right? So we all agree on that. But what is data diversity? That's the first point. When you think in diversity across level of the brain, you have a lot of diversity that is beyond the classical categories like gender or race, right? So how you want to measure brain health? Like you want to use neurotransmitters? Do you want to look at the local interactions in the brain? You want to look at, hemodynamic responses, et cetera, et cetera, et cetera. So we don't have any kind of systematic approach to linking different layers in the brain. But also, we neuroscientists usually forget very quick that the brains work for the body and not in the inverse situation. And bringing body, especially, inflammatory, immune, metabolic pathways is critical to understand brain health and resilience. And then connecting the body with the environment, like connecting, physical exposome like pollution, for instance, or social exposome like socioeconomic inequality. For this, we really need to bring whole body health, exposome, and brains integrated, right? So that's a kind of extended definition of, if you want, of diversity or heterogeneity in data. And for that, you need really different computational models that really embrace heterogeneity, that are able to, integrate different levels of information and to reduce to multiple, high dimensional manifolds in low dimensional representation of this. And I will come back to this idea later. And the other thing is this is important for both psychiatric and neurological conditions, because when you try to integrate this level from genetics, epigenetics, whole body physiology, exposome, lifestyle, so many factors are cross-dimensional, transnosological, and I think that we need to move into that direction in order to develop better models. But there is a more deeper why we need to bring this really global diversity approach to understand resilience and brain health. Let me give you some examples. For those that are, working in the field of dementia, you will absolutely know these graphics. It's the Lancet Commission. So you have 14 factors that are there. Most of them are modifiable, and you can, reduce up to 45% the risk of dementia if you will be able to, control these factors. But for example, just thinking in poverty or socioeconomic inequality, or, wealth shocks. Wealth shocks are things that we are very familiar nowadays. We just did a few modeling of this, and, the number is huge. So the socioeconomical conditions and many other conditions beyond the classical risk factors really call for a different agenda, not only, involving these different features. There are so many others. The problem is we needed really to bring the body and the exposome on that. And this picture was made by my friend, Johnny Miller, here in the right part. Johnny was a very unknown photographer in South Africa, originally from Seattle, from US. And one day he took this picture and then became extremely viral, famous. Now he's, presenting his work across the globe. But the tricky thing is, if you're born on the, right or left side of this picture, your socioeconomic life is going to be completely different. So this work of Johnny really inspired me to show that it's not only a socioeconomic problem if you're born on the left or the right. It's also a whole body health problem, and it's also a brain health problem. So if you live in the more deprived side, your brain will have to deal with sustained stress. So the allostatic interoceptive network of your brain will be more dysregulated. You will have dysregulation for the cardiometabolic, cardiovascular system. You will have, higher risk across cardiometabolic, metabolic, vascular, immune parameters, right? And of course, your ability to self-regulate and the kind of structural, infrastructure supporting your health, it will be weaker. So this is the why. In this paper, I just published that, because Colombia represents an extreme case of what happened in the world. Colombia is facing an incredible pandemic that includes displacement, massive displacements, increase of violence, narco trafficking, huge socioeconomic inequalities. But I'm pretty sure that many of us will recognize these kind of challenges that we have in the future ahead. And if we don't bring brain health into the real, kind of factors, environmental factors that impact our health, I think that we are going to be very poor in developing precision brain health models. And this is why I think that it's so important to have a more ecological view of brain health and resilience. So I will summarize some of our ideas that we published in this paper recently as part of the 30 years of Nature Medicine anniversary. And the first things that we want to bring is, resilience. It's really dependent on different pathways: biological, cognitive, psychological, and social. Mechanisms of resilience are, very, they can be short span or very large span. But the most important thing, there is nothing as a simple definition of resilience or simple measurement of resilience. So resilience accumulation is a combination of different genetic, cognitive, cultural, environmental, social, and brain factors. And we need to develop better methods to understand that. We already know that you can identify resilience across this level. Just let me give you some examples. The one that I am so proud to share, is that the most important resilience aspect at genetic level for Alzheimer's disease comes from Colombia. So there are two genes, the colbos and the Christchurch variants, that really provides a protective effect, meaning that even if you have the presenilin-1 mutation that induces dementia, you won't develop the disease, right? So this represents a unique opportunity for genetical trials, and this is an extreme case of resilience at genetic level. But environmental resilience is also important. In another paper published in Nature Medicine with more than 160,000 participants, we assessed how the exposome really modulates accelerate aging using deep learning and machine learning approaches. And we found that many environmental factors can induce resilience. But another important thing is community resilience. I think that many of you will agree that the cumulative impact of stress is a really risk factor for, cardiovascular disease, cardiometabolic disease, and also dementia, right? What happens if your social network or your family helps you to manage the stress across the lifespan? Probably you will be more resilient for that. So I think that it's time to realize that there is nothing as a resilience defined at a single level and in a static point of view. So we really need to bring a spatiotemporal richness of resilience if we want to develop more robust assessment. So by saying that, I will bring some examples how we can bring whole body health and exposome or environmental factors to try to understand this better. So the first thing is, when we talk about diversity, most of the time we talk about disparity, like early diversities, low socioeconomic status, pollution, low education, gender disparity, cardiometabolic conditions, right? And for that, we really need more global data, because all these factors, they especially impact the body and then the brain. So cardiometabolic conditions are the most simple example of how inequalities or disparities can really create huge impacts in your body. 2 months ago, there was a wonderful paper published in Nature in animal models, and in which they really demonstrate how social stress can induce a typical response in the amygdala, that influence the glucose in order to, having better adaptive response to environmental demand, and then you can really induce diabetes. this kind of examples show how social factor or environmental factors can impact your body. But we need to move forward and understand how the body then impacts the brain and how they are allostatic interoceptive overload loops, how these effects in the body impacts the brain, but at the same time, the brain then impacts in the body through the continuous responses to environmental demands. And this is the other thing that I have been trying to push, this idea that for understanding the connections between environment and brain, we need to bring the body, right? From physical exposome, including pollution, heavy metal, toxins, plastic, and more important, nanoplastics, to social exposome like structural inequality, educational disparities, socioeconomic status or stress. All of this impacting the internal responses, internal biological responses, like, epigenetics, allostatic overload, inflammation. And when this is, sustained in time, the risk for multiple disease becomes more accentuated. And that's the point where we need to understand resilience. What makes the people, living with the stress in a different way? How may the peoples or the body of the people, to don't regulate the environmental demands in different ways? Once again, for this, we really need to understand the impact of environment, not only in the brain, but, in the heart, in the lungs, in the viscera in general, even in the musculoskeletal system. This can be simplified through the allostatic interoceptive models that, brain prepares the body for responding to environmental demands. And this is a way to adapt. You have more energy, you reduce the immune system, you increase inflammation, and you can adapt to environment. And this create, kind of allostatic load where you have adaptive responses. But, when these external factors increase and are sustained on the time, these chronic exposures to stress create these adaptive responses. And then you have, kind of allostatic overload responses where hormesis really create an overreaction. And this difference can mean, resilience or non-resilience organisms. And for that, we cannot just understand the organisms, we need to bring environment once again. The other point important for resilience is how your brains self-regulate your body into environmental demands. From physical demands to social demands, we know that the body reacts to stress and reacts through inflammatory pathways, right? And across multiple loops from interoceptive visomotor and specific brain regions. So we have been working with frontotemporal dementia to try to understand how interoceptive signals really drive many of the symptoms and manifestation of the disease, but this, I think, is beyond frontotemporal dementia and is a standard way to understand how environmental demands requires adaptive or resilience responses that in specific conditions can become pathological. With George Northoff, we have started to create computational models to measure interoceptive signals from different visceral signals and then trying to use that to identify as predictor for clinical outcomes. We also have shown with frontotemporal dementia, for instance, that these interoceptive signals are associated with clinical, cognitive, and also brain atrophy signals associated with the disease. And more than 5,000 meta-analytic approach to different studies, we have shown that deficits in brain region associated with interoception are always associated with emotion and social cognition process, bringing once again the body, in the regulation of cognitive process. And in this paper just published a few days ago, we shown that, the allostatic interoceptive network, this region of the brain that is regulating the responses to environmental demands, become impaired when it's associated with unhealthy body health. We use that, cardiovascular scores, and we've shown that the larger the cardiovascular risk, the lower the functional connectivity and the structure of the allostatic interoceptive network in the brain. But this is not only the only way to look at the the cardiovascular system. Probably some of you are familiar with the work of Tian. She has made an amazing job showing how, you can better predict health and disease in the brain using information for more than 3 organs than using just the brain. I think that this is the most wonderful demonstration how, in this case, using organ clocks metrics, but how the organ clocks metric, how the health metrics from the body are extremely relevant to understand health and disease. And we need to move from just measuring the brain for that. Then let's move from the whole body to the exposome. And let's start with the social exposome. By definition, the social exposome is any environmental factor that requires others and that can be associated with health outcomes across the lifespan. So starting for education, but also, your social interaction, your socioeconomic status, your access to your health system, let's say, how is the exposure to violence? How was the exposure? How was your childhood? All these processes are part of the social exposome. Starting for a very, poor metrics. The socioeconomic inequalities is a metric that the economy is well known, and it's a very poor metric. But structural inequality, it's better associated with brand signals than, age, gender, cognition, and education factors, showing that there is a huge modulation of the basic metrics that we have to understand brain health associated with these metrics. In this another study, we compared just the structure and function of the brain of Latinos versus US in aging and dementia. And we have huge difference, right? So a large percentage of the variance of this difference was associated with years of education, these very poor and simple metrics. But when we look at qualitative measures of education, not just years of education, the amount of brain reserve, the amount of brain volume and functional connectivity that is explained by education is up to 7 times larger than just years of education, pointing at the importance of what we learn, what is the quality of your education. In this other paper published last month, we decided to go further and collect more than 200 variables associated with the social exposome. This variable included metrics of education from, not only years of education, not only quality of education, not only if you were in a, yes? It was a question there? No, sorry, I thought, okay. So education across multiple levels, education measured not only at the level of the participant, but the parents. Then, how was the food insecurity across the lifespan, the financial status across the lifespan, the access to healthcare, it was child labor in the past or not, the subjective and objective metrics of socioeconomic status. And we created a kind of cumulative burden with these hundreds of variables. And this cumulative burden was truly associated with not only, cognitive level, but also functional ability, also neuropsychiatric symptoms, and also, brain research in terms of structure and functional connectivity. This happened in healthy controls, in Alzheimer's disease, and frontotemporal dementia patients. So this is another study that we'll skip. But then the next question is, can we understand how different, let's say, physical and social exposomes interacts to shape brain health and disease? So in this first paper, we started to use brain clocks, and we started to look at the people that have a younger, a resilient biological age of the brain versus an older age of the brain in comparison with the chronological age. For that, we use multimodal neuroimaging and deep learning analysis of convolutional neural networks, and a little bit sophisticated metrics of connectivity, high order interactions that capture more than bivariate connections in the brain. But the simple history is that we created this model of brain aging, and that we shown, it was very interesting. So first of all, people in the South tend to have accelerated brain aging. Females tend to have accelerated brain aging. People with dementia tends to have accelerated brain aging, right? But if you are a female coming from the south and having dementia, you will have the largest brain aging, the larger gap with your chronological age. And one thing that we started to explore that was both pollution, that is a critical component of the environmental exposome, and socioeconomic status, that is another component of the first one is physical, the second one is social exposome, were associated with this accelerated brain aging. But then in the follow-up study, we developed a more simple biobehavioral age gap, like estimating the age of the people, just getting information from, risk factors like heart disease, hypertension, hearing impairment, sensory problems, cardiometabolic health, also, how strong were your physical abilities, your education, well-being, functional ability, and cognition. With that, we were able to, using, large data set, in these cases, 160,000 participants, using matchings to estimate the kind of biobehavioral age. And the first thing that we found is that the people that have more accelerated aging comes from Africa, from Egypt and South Africa, followed by Latin America, followed by Asia, then Mediterranean countries, and then the northern countries, in this case, Sweden and Denmark, have the more healthy, less accelerated aging. But the most important thing is we discovered that income, socioeconomic inequality, gender inequality, pollution, or even, political instability measured through aggregate level democracy indexes were inducing accelerated aging. Once again, bringing this idea that your age or the pace of your biological aging doesn't depend on genes or biological process alone, but the environment plays a very important role. In this follow-up study, now in second review in Nature Medicine, we expanded the number of exposomes to 72, including multiple metrics of physical exposome like green space, temperature, precipitations, climate disaster, disease-related mortality, quality of soil, water, air pollution, also social exposome, structural inequality, gender equity, unemployment, GDP, and democracy indexes, and as well as another metrics like migration. That we have is all of these individual factors, called exposomal factors, have an important effect in these lines that I am marking, associated with inducing accelerated brain aging. But when those factors were combined, it was a quasi-exponential effect in inducing accelerated brain aging. This is by definition the exposome, right? So the exposome is like the genome. It's the composite, the combination, the compound force of environmental factors inducing accelerated brain aging. We've shown that, when you have more than 10 top predictors of exposome, the likelihood to have accelerated brain aging was even larger than having a clinical condition like dementia, like Alzheimer's or frontotemporal dementia. Now we are starting to understand what underlying mechanisms are associated with accelerated brain aging. So the first thing that we did is assessing, very fundamental biophysical mechanisms like the excitatory inhibitory balance, synaptic plasticity, and global coupling of mesolimbic networks. And we've shown that these very fundamental mechanisms that are all associated with unhealthy aging are markers of accelerated brain aging. I just say that because now from these two studies, we are studying exceptional participants that have an extremely younger biobehavioral age or an extremely younger brain regarding the chronological age. We have like 20,000 participants with biobehavioral younger ages and 3,000 participants with really younger brains. We already confirmed that there is no artifact associated with that, so it's not an error in the matrix. And we are just starting to explore which factors drive resilience. It's just a preliminary data. We don't have nothing definitive yet, but the first thing that we discovered is that there is not one single factor. You have to have multiple combinations of protective factors to starting to have a very, very delayed brain age gap. And also, I don't want to overemphasize the environmental factors. I think that the genetic factors are essential. In this work, we use Down syndrome as a hallmark of accelerate brain aging condition and compare it to genetics and sporadic versions in Alzheimer's disease. And that we found with a very, very complex computational pipeline is that the Down syndrome have up to 20 years of accelerated aging, that the accelerated aging start far beyond the estimated age of onset, while, in genetic presymptomatic presenilin 1 mutation start about 6 years before the age of onset, that this strong accelerated brain aging is associated to, intrinsic excitatory inhibitory mechanisms with, more expression of accelerated brain aging across different genes associated to Alzheimer's in general, but also some specific markers of Down syndrome pathology. Then we use biophysical models and perturbation models to understand how the disbalance in terms of neuromodulation and drug delivery can reverse the accelerated aging of the brain. So this is a work that is under review, but I want to bring in because now we are starting to understanding how genetic resilience can induce and can impact in accelerated and delayed brain aging, And I just want to mention that how much I admire Francisco Lopera as one of the most important geneticians that have been working for discovering not only, he discovered the largest kindred of presenilin 1 mutation in the world that has come from Colombia, but also he discovered these two critical genes, the Christchurch mutation and the Colbos variant, that, stopped the genetic presentation of a dementia from progression. And now we are just starting to understand in the people that carry these variants, how these mechanisms induce resilience even when they have a presenilin 1 mutation in parallel. So I think that for understanding resilience in brain health and brain settings, we need to move beyond the individual level variability and bring the environment. And then measuring across different multi-aging clocks, that is important. And I think that we need to stop thinking in unidimensional aspect, like in more environmental or more genetics. We have papers that compare the impact of environment versus genetics as they are separate factors. They work in a very blending way. There is a really blending of exposome and biological or genetic factors that we don't understand, not only, let's say, through social epigenomics, methylation, and other factors, but developing really more complex computational models to capture both genetics and environmental factors to understand resilience and vulnerability. I think that this is far beyond brain health. Disease of despair, like suicide, alcoholism, and substance abuse, are rising in many parts of the world. And we usually have been understand that at the individual level, what's happened at individual level. We need to understand macro level predictors, like how these exposomal factors impact that. We are going to develop some global projects for that, but also to trying to bring resilience into the table. I think that studying accelerated brain aging is important. So for understand resilience in this paper, in 27 countries, we shown that multilingualisms can really delay accelerate aging, with a strong effects, even controlling for multiple languages and environmental factors that never have been controlled altogether in the literature. So there is a strong effect of 2, 3, or more language in, protecting against accelerated aging. And in this paper that we published last week, we've shown that different creative activities, like tango dancer, musician, visual artists, or even gamers, playing specific games that require, strategic thinking, imagination, and flexible decisions. All this process, all these people that are experts or having more creative experiences, they systematically have delayed brain aging in comparison to non-experts. And we also shown this that the training effect can have a direct effect in the pace of brain aging. And we also exploring some biophysical mechanisms associated with that, but I don't want to burden you. So I think that we really need to develop a better understanding of how environment, whole body, and brain interacts. The Click Brain is a project that I have the great pleasure to launch in a couple of months that will try to work with transdisciplinarity, like, integrating people that measure pollutions with satellites, with people with behavioral neurologists or computational neuroscientists, to developing innovative metrics to connect environment to body to the brain. I think that this is something that we really need. As you may know, the largest the transdisciplinary flavor of a project, the less likely to get founded. There are very nice tragic results showing that, the transdisciplinarity, even when we all try to promote that, doesn't work well in mainstream settings. So I think that we need to bring more transdisciplinarity. And just to try to close in the remarks about resilience, it's not just that the resilience is a multimodal factor, right? That you can have resilience at genetics or whole body or community or even large environment like exposome levels. The problem is beyond that, is that we don't have really integrated metrics of resilience. So traditional resilience scales are good, but they measure just psychological demands. They are, limitations, self-report, validation across cultures are not good. Then we have the WHO intrinsic capacity framework that have a, more holistic assessment, combining, physical exams, cognitive tests, self-reports. They are more applicable, but, requires larger resources to measure that. And it really needs adaptation in different cultures. Then you have cultural adapted tools. They are, well adapted for specific cultural settings, but at the same time, they are very time consuming and there is no standardization for comparisons. Then you have, more computational biological frameworks to assess resilience, that you can bring aging clocks or responses to stress, physiological measures, but that requires much technical expertise. They are not accessible and they are more hard to harmonize across settings. And more recently, we started to have wearable technologies, right? So you can measure real-time physiological parameters like heart rate variability or other physiological responses to stress to identify, shortcuts to resilience. And you can have real-time monitoring and more temporal assessment for that. But of course, there are multiple problems, from privacy concerns to engagement to technological infrastructure and other problems. So I think that the first step is to really create a theoretical model that can bring resilience in the whole complexity that we need to address, and then developing some metrics. I just leave for you. I won't go into the detail to don't overextend my talk, but we have suggested a roadmap for promoting brain health and resilience that brings this idea that for resilience we need to understand different levels, like cultural practice are important to understand health strategies. That educational access, it's critical for the classical approach to cognitive and brain reserve. That we need to focus in a structural aspect, like poverty and socioeconomic inequalities we cannot ignore that. We just cannot be based in individual behavioral change because the degree of freedom for that is very small. There are, of course, practical recommendations like nutrition, dietary practices, and the other important thing is if we want to develop measure of resilience, we need to go into the local context. We need to measure specific factors across levels that are associated with resilience in a specific context. Of course, interventions across life and multi-sectoral collaboration and policy integrations are really important if we want to bring resilient measures outside the pure research or biomedical settings and trying to bring them as an important policy approach for recommendations. So that's my general ideas about resilience, and happy to chat with you more. Thank you so much for your attention. I don't know if there are any questions, if you want to comment on something.
I have a question. This is Sean Savitz from Houston. Thank you so much. I really, really have enjoyed this lecture, and there's so many of your publications that we as a group need to be, reading, and you're providing so much new information over time, and so we were so happy you had some time to give us today. When I think about where all this data will go, I was wondering about how do you stratify the bigger contributors versus protectors?
Well, I wish that we could do it. We are very far away from that, but you are pointing a very specific point. So I can just give general insights about that. First of all, there are some few factors that can be considered like general, right? So but those factors doesn't have a single solution. So what is the most, across all studies, what is the most important factor that drives risk is poverty or inequality. So that's a big issue, right? The other thing is developing tailored measures, because even when you have these big giants playing an important role, the level of individual variability is huge. It's much larger than we understand. So the only thing that I can mention about that is we need to develop tailored models. These tailored models need local data. So we need to really don't just extrapolate models from other regions, but having our own local measures and developing computational models that capture heterogeneity. We use a lot of data augmentations and probabilistic metrics to embrace heterogeneity instead to trying to, ruling out from the models. So addressing heterogeneity is crucial to understand individual trajectories and, of course, resilience. Then I will say that there is a very black hole in the literature, that is how environmental factors interact with genetics. We don't know almost nothing about that. Of course, we have some measure of methylations, right, or some polygenic risk scores that you can combine with polysocial exposomic metrics, but this is a very simplistic way to understand the huge blending between environmental and biological factors. So I think that we need to work on that. And the last point, I think, is cardiometabolic health. I think that cardiometabolic health is the perfect link between, first of all, inequality and environmental factors, right? But then brain health. I work in the field of dementia, where vascular components has been neglected by, the diagnosis. So if you have vascular component, you have vascular dementia, but it's not part of dementia. Nowadays, we know that this is a completely wrong way. So from the vasculature of the brain, that is directly associated with genes that are, risking for dementia, to this, we were starting to talk with Fadi about cerebral small vessel disease, perivascular space, wide matter hyperintensities, microhemorrhage. But then also, the connection between the glymphatic system, the vascular components, and dementia are so clear. So if you ask me, point to one aspect that is not environmental or is not directly environmental, and you can tackle it, I would say cardiometabolic health. We really need to bring the peripheral cardiometabolic health as a core component to understand resilience and individual variability. But they are very, not really precise answer, I'm afraid.
And can I ask you your personal view at this point on air pollution? Since this is such a huge problem, Houston's air quality is terrible in the summer, for example. I'm thinking about urban versus rural centers. So since your population density is obviously on the urban, many of us can do lots of things. We can engage in physical fitness, diet, control our cardiometabolic factors. So I really wonder, in your view, how much does air pollution play a role for us? I mean, should we, as we get older, think about retiring to the mountains? Is there a certain amount of exposure to air pollution where we should basically, I hope your data will eventually show, or somebody will, that, there's a certain amount you should be exposed to before it's time to pack up the bags and move.
Yeah. So it's another wonderful question. I think that pollution across some models that we have tested with different data is one of the most systematic factor, is one of the largest effects that we have found, depending, of course, the impact, not only in unhealthy aging, but especially in dementia. So this is one point. The second point is, why we talk about pollution. Most of our measures are really poor metrics, like particles, like PM2. This kind of metrics of the size of a particle doesn't provide much information about the specific particles. When you look at that, they are huge. There are some evidence, not from my group, that showing that the specific type of particle have really harmful effects that we need to address. And then the combination of nanoplastic with pollutions is a very well neglected process. It's hard to get information, but we know that nanoplastic, goes through the brain, but we don't have really efficient ways. Now we have some wearables that can measure the air quality, for example, and I hope that we can develop better precision models. But the problem for moving, to establish a threshold, it's twofold. So one is we don't have enough granularity of the metrics of pollution in time and in space. It's both. So our metrics are basically using the WHO metrics that you provide for a region, for a year, and this is like a picture, right? It's nothing dynamic. But then, what are we really measuring in air quality, for example, or in comparison with water quality? So in Argentina, for example, we have a region with the largest mortality and largest comorbidity, and we have an incredible amount of heavy metals in the water. So this is something that we don't have really good approaches to measure that. This is one of the problems. The other problem is what to do. As you say, so we need to pack it and leave. But at the very least, I would say that there is evidence that the mitigation actions, like, just covering this part of your face, or, not doing exercise with some peaks of pollutions, can really have a mitigation at the very least. That I think that is important. But once again, I think that we are very far away to having a more precision approach or providing more specific cutoffs.
Thank you. Really great presentation. This is Fadi again. So I have a question for you, and this is probably very basic. So, you read in the literature, there is resilience, there is reserve. So what's the difference in the model, if you're modeling for reserve versus resilience? Is it different? Is it the same? So not too much work, and everybody tried to say different definition based on what they think is the reserve and resilience. So if you can, just say some words about those differences.
Yes, it's a great point. In our Nature Medicine paper, we were asked about that, and we were fighting against us between the co-authors because we didn't come up with a standard definition. Between the authors was Jacob Stern, one of the most important researchers on cognitive reserve. So I will give you my opinion. I think that when we talk about reserve, we talk about something more static, something that comes from the past, that you already have it. And you can talk about cognitive reserves of maybe education, or you can talk about brain reserves, so you can measure the volume of your brain or the functional connectivity of your brain. While in resilience, you are pointing more at a more dynamical concept. It's really changing. It's really more multidimensional. And it's also directly associated with adaptive response to disparity. I won't say that this is always the case, but in general, resilience, it's, better illustrated when you have, let's say an environmental factor or a personal life challenging situation, and the way that you keep yourself healthy and you are able to respond to environmental demands. This is more resilience than reserve. That should be my distinction, but these are a really gray zone with no agreement across many authors.
Yeah, thank you.
Thank you.
Okay, I don't see any other questions actually, and we're right on time. So thanks again really for joining, and we'll be in touch, of course. So we're also putting really, we're working on the reserve also. So hopefully if we get, towards, we'll happy to collaborate, Yeah, thanks.
Yeah, I was going to say, that the collaborations you're doing in Latin America and the country, if there's something we could be doing here in Houston and Texas to be working with you, we'd love to.
I would love to, especially because you have many Latinos, and comparing Latinos that have been, migrating to US or are second generation versus Latinos in their own setting, it could reveal a lot of resilience, a difference between biological, genetics, epigenetics, environmental factors. So I would love to explore collaborations. Also, I would love to explore with Fadi these ideas of cardiovascular clocks that we are developing. I didn't show nothing about that because it's quite preliminary, but we are working on that, and I would love to explore that. And yes, I will be delighted to explore this collaboration. We have 7,000 participants, aging, Alzheimer's, and frontotemporal dementia, very well deep phenotyped, and we are really, really looking for exploiting this data set and making comparisons. So more than happy to exploring data sharing or collaborations.
There are several colleagues on the call. They're from South Texas. We have West Texas, in different Hispanic populations, different ancestries. So I think that could be really interesting to consider collaboration in the future.
Wonderful. Anytime. Thank you so much for the invitation and for your interest. I am looking forward to explore collaborations. Yeah.
Yeah, thanks.
Thank you.
Thank you.
Thank you very much.
Thank you. Bye-bye.
Bye-bye.